guys, in the last session we have studied about PCR, polymerase chain reaction. It is used to amplify the gene or amplify the DNA. So here you can understand we have to isolate DNA. It is cut into the fragments and it is separated by the help of gel electrophoresis. So and it is inserted into the plasmid. That only you can understand about the rDNA. rDNA. Now, this rDNA or recombinant DNA, we can select by the help of selectable markers. Already in the previous videos, we have studied what are the selectable markers. So, by the help of selectable marker, we can select only the recombinants and we can reject, we can eliminate the non-recombinants and we can permit the growth of only the recombinants. So that we can say recombinant DNA. Now the next step we can understand foreign DNA, obtaining the foreign genome product. So we have to get obtaining gene product. So actually by the help of this biotechnology rDNA, we get the gene. But this gene, it has to convert into the product or the protein, biological protein. Then only it can express the character. So we have to, this gene we have to convert into protein. Then it can inherent. It is inherent and it can express the character. So that's by the help of one of the instrument. It is called bioreactor. It is called bioreactor. So what we can understand, the gene, biological gene, we have to convert into product or the biological product. That only we can say protein. So that's done by the help of the instrument which we can say bioreactor. And we need large amount. That also by the help of this bioreactor. Here you can understand. So by the help of bioreactor, we can produce large number of biological product. We can convert the biological product. So that's by the help of bioreactor. There are two different types of bioreactor: simple stirred tank bioreactor and tank bioreactor. But mostly in biotechnology we are using simple stirred bioreactor. Here you can see this is the arrangement of simple stirred bioreactor. Here by the help of this we can make large amount of biological product and it has the facility to produce large amount of biological product. So next we can see what are the facilities it provide to produce large amount of biological product. So here you can see sterile air, always it provides the air. So it is used to stir the biological product. It is used to mix the biological product, as well as here you can see the curved base. That also we can see it is used to mix, it is used to stir the biological product. And here you can see when this is the motor, when the motor runs, immediately the blade it will be rotated. So when the blade is rotated, immediately the foam will be formed. That foam should be broken. So that you can understand, blade it is used to break the foam. And again acid base controller, or you can see here pH controller is there and steam sterilization is there. So this provide optimum condition, optimal growth condition for the biological product. So that we can understand about the advantage of this bioreactor. It provides optimum condition, optimum pH, optimum temperature to produce large amount of biological product. At last process you can see here, last process, it is called downstream processing. It is called downstream processing. So what's this downstream processing? Still we are using this bioreactor. We got large amount of product, but that biological product or protein is contain some impurities. So first of all we have to remove these impurities. We can remove the impurities by the help of different separation process, purification process like decantation, sedimentation, chromatography. Using all these techniques, separation process, we can separate, we can remove all the impurities it contains this biological product. So after removing these impurities, that process only we can say downstream process. After removing these impurities, we have to add, we have to formulate with suitable preservatives, we have to use preservatives. Then after the using preservatives, we have to pack properly this product. If we are not packing this properly, again it will be damaged, so spoiled. So we have to pack properly. Now it is ready to send, it is used, it is ready to send to the market. So after that we can send to the market, it can be used. So this is the last process, the process of biotechnology. So here the chapter biotechnology principles and process, it is over, it is finished here. Now I will upload some shorts, it contains some neat recent PYQs, recent 24-25 recent PYQs. Please listen those shorts and please reply your answer in the comment box. And if you have any doubt regarding NEET, this chapter biotechnology, you can comment, you can ask in the comment box. So I expect your coordination, cooperation, and I expect you all people will listen my NEET questions. I expect the comments also. Yes, thank you students.